Hello all, myself Priyadarshini, Assistant Professor, PG and Research Department of Biotechnology, JJ College of Arts and Science. Today we are going to discuss about the cryopreservation. Cryopreservation. It was introduced in the year 1866 by the Russian chemist scientist Alexander Zeister. Here cryopreservation, usually in the topic itself, the meaning is shown. Cryo is a Greek word. It represents cold preservation. The meaning of the cryopreservation is cold preservation. Cryopreservation is a non-lethal storage of biological material at low temperature, very low, extreme low temperature. At the temperature of liquid nitrogen minus 196 degree, almost all metabolic activities of the cells are ceased, and the samples can then be preserved in such state for the extended periods. That is a prolonged period of the time, long-term storage. However, only few biological materials can be frozen to minus 96 degree without affecting the cell viability. Actually, in this point, what they have said is we can't able to store all the biological materials. Only few can be undergo to the process of the cryopreservation at minus 196 degree Celsius. This cryopreservation will be done without affecting the cell viability. Cell viability means the cell that has to be survived. That is, cell viability means the survival of the cells. Liquid nitrogen. Liquid nitrogen is mostly used material for the cryopreservation. Sometimes the dry ice can also be used. Why we are using the liquid nitrogen? It is chemically inert, relatively low cost, non-toxic, non-flammable, readily available. So that we will be preferring the liquid nitrogen. And we have to know what is cryoprotectant. Cryoprotectant is a substance used to protect the biological tissue from freezing damage. You know, while we are freezing the cells, it will undergo into the process of the damage to the cells. So, we have to protect the cells from the freezing damage. We will be adding some cryoprotectant. That nothing but protecting the cells. Example, glycerol, which protects the blood cells and the bull sperm, and DMSO is a dimethyl sulfoxide. It's used for protecting the cord blood cells. These the two cryoprotectant popularly we will be using as a cryoprotectant. Sometimes salt along with the ice cubes, salines can also be used as a cryoprotectants. Function of the cryoprotectants is it prevents the crystal forming inside the cell during the freezing process, and also it protects the cells from death. Process of cryopreservation. First of all, we have to select the material, that is harvesting of the material. Here we have to check the material's volume, its density, its pH, its morphology, and whether it is damage free or not. Everything we have to check. That is the selection of the material. Second thing is, after the selection of the material, we have to add the cryoprotectant to it for the protection, and then we shall go for freezing. Different methods of freezing is there. Sometimes along with the freezing, some warming will also be, sometimes we can expose to the warming also. And third is thawing. Thawing is the process of warming the samples in order to control the rate of the cooling and protect the cell damage. Here it's the warming process. Why it is need is for the control of the rate of the cooling. It controls the cooling. Our cell should not be in a very low temperature. And the last is determination of surviving and the viability. It viability means whether the cells is survived or not, we have to check. The cryopreserved samples are stored in minus 80 degrees Celsius in a freezer for about 5 to 24 hours before transferring into the storage vessels. We have to check. We have to do minus 80 degree Celsius in a freezer for 24 hours. After that, we can transfer into the container. Here, first thing is storage. The frozen cells or tissues are kept for storage at the temperature ranging from minus 70 to minus 196 degrees Celsius. The temperature should be sufficient low for long-term storage of cells to stop all the metabolic activities and prevent biochemical injury. Long-term storage is best done at minus 196 degree Celsius. And thawing. It is done by putting ampule containing the samples at a warm water bath 35 degree to 40 degree Celsius. The frozen tips of the samples in tubes or ampules are plunged into the warm water with a vigorous filling action just to point of ice disappearance. It is important for the survival of the tissue that the tube should not be left in the warm water bath after ice melts. And this is a liquid nitrogen cylinder to which the cells are stored. And this is a short term storage of the embryos. The embryos have to be stored for some 2 days or 3 days, means we shall make the embryos to inoculate into the medium with a saline along with the cryoprotectant in a petri dish, and we can store it for 2 to 3 days. The embryo will be viable. And the determination of the survival. Regrowth of the plants from stored tissues or cells is the only test of survival of the plant materials. We have to check after the storage, we have to check whether the stored cells or survive or it is in the death phase. So that we have to determine the survival. Various viability tests are there, like fluorescent diacetate, FDA staining, growth measurement by cell number, dry and fresh weight. Important staining methods here are Evans blue staining. Let's discuss what is Evans blue staining. After the taking out of the cells from the process of the cryopreservation from the liquid nitrogen cylinder, we have to check whether it is live or in a dead state, the cells which is in live or in dead. So we have to add 0.1% of Evans blue reagent into the cells which is in the microscopic slide and observed under the microscope. We have to observe under the microscopes. Only the dead cells, that is non-viable cells, only the dead cells will uptake the Evans blue, which looks blue in color. The live cells will not uptake the dye. What's happening? What's the mechanism there? It's the dead cells plasma membrane will be broken so that the dye that can easily enter into the dead cells. So the dead cells uptake the blue color. The live cells plasma membrane will be as such, so it will not allow the stain to entering inside the cell, so it looks very normal. The blue color cells which are dead cells and the white color cells which are in a live state. Percentage of the viable cells. Number of fluorescent cells into 100 by total number of cells. This viable plus non-viable cells will be counted. The last thing is application of the cryopreservation. We can freeze the cell cultures and maintenance of disease-free stock for the maintenance of the long storage of the seed varieties which can be um, used in the future. Gene bank, the genes can also be stored, and the storage of rare germplasm resources. For that, cryopreservation that plays a major role for the storage of these important things. And this is a plant biotechnology. In case of animal biotechnology, we can store the embryos, sperm, semen for the long term storage. After that, after some 10 or 15 years after, we can take out the embryo or sperm which is in a live condition without losing its metabolic activity. Thank you very much.